Our guest today is Dr. Jose Leme. Jose is an assistant professor of cannabis biology and cultivation systems at Southern Illinois University, Carbondale. And he runs a lab studying how environmental controls, nutrition, plant hormones influence cannabinoid production and trichome development, among other things. And what makes Jose particularly interesting to talk to is that he didn't come up through cannabis. He came up from precision agriculture, corn nutrition, and satellite remote sensing. So he brings a level of agronomic rigor to cannabis that the industry hasn't had much access to. And I think that the cross-pollination between traditional agriculture and cannabis science is where a lot of the most useful insights are going to come from for the field going forward. So his lab has published a series of papers over the last several years that I found really interesting, with some surprising findings and some results that challenge some of the assumptions that most growers, myself included, have been operating on. So we're going to get into some of those today. So Jose, welcome.
Thank you. I'm very happy to be here talking to you. I think it'll be a very interesting conversation.
We're really excited to have you here. So let's talk a little bit about trichome findings. You did a study on methyl jasmonate that found something like 32% more trichomes, but the CBD and THC didn't move in the results. And the untreated controls, I think, had the highest CBD. What do you think is going on there?
Okay, so just before answering this question, just talking a little bit about my background. Yeah, in fact, my background is in agriculture. So I did my bachelor degree in university in Brazil, Londrina State University, and in agronomic engineering. Then my master's degree at Auburn, I worked with precision agriculture. And this is interesting because a lot of the techniques or the technology from precision agriculture is being transferred to control environment agriculture and vice versa. So, but I guess precision ag is just more conventional farming, and then control environment agriculture is more horticulture or indoor farming. And then during my PhD, that's when I started working more specifically with cannabis and indoor cultivation and the production of plant secondary metabolites. So, okay, talking about your question. So, yeah, so this methyl jasmonate study, it's actually very interesting because we conducted this study in 2021 and 2022. I would say that it was after the Farm Bill, the green gold rush, that everybody started growing high CBD plants outdoors, and everyone trying to chase that green gold was just after, but the outdoor production is still there. And so that's why we decided to conduct that study in outdoor setting instead of indoor. In most of the cases, I prefer to do indoor studies because I guess I'm biased and I'm working in control environment agriculture. And I think when you do indoor studies, when you conduct indoor studies, you have a more like a cleaner data, a more straightforward data. When you grow outdoors, obviously you have much more variables. But anyway, this study was outdoors. And I would say that this was the first study that we start seeing the trichome density not being directly correlated to cannabinoid content. So we can talk about that later, but more specifically talking about the effects of methyl jasmonate in trichomes. So basically in this study that we're talking about, we apply methyl jasmonate and also we apply pesticide because, as you know, methyl jasmonate is a plant hormone that the plant release when the plant feels that they're under attack, that it's something going on, it's a stress happening. So, and then naturally the plant release that plant hormone. So, and then in order to avoid confounding the results, we decide to include one treatment where we are applying pesticide. So our idea was, or our assumption was, not having the natural production of methyl jasmonate due to the pest attack, because we'll be applying pesticide, will be a better way to actually see how much effective is methyl jasmonate. So then we got the results, and in fact, we found that it's increasing trichome density, but that was not correlated to cannabinoid content. So again, I think if you think, I mean, now there are more studies addressing the effects of methyl jasmonate in cannabis, and it makes total sense because it can be controversial or not. But I mean, you know what I'm saying that because there are people with very different opinions about that. But more recently, we start seeing some studies showing that the trichomes are structured for that are formed for as a defense mechanism. So
Mhm.
pretty much to protect the reproductive part of the plant. And then if you think go and if you talk about biology, it's kind of it can be a crazy thing because it's pretty much like little hairs or little biofactories that are protecting the reproductive part of the plant. And when we talk about human beings, also we also have pubic hair that is also to protect the reproductive part of the human being. So anyway, not going too far, going back so to cannabis. So if this is a defensive mechanism, why not artificially apply a compound or a plant hormone that is naturally released when the plant is under attack or when the plant is under stress and create that fake signal?
Mhm.
Oh, you are actually under stress, but you're not under stress. So it makes total sense. And when we decide to conduct this study, we thought that the result would be more straightforward as we thought for all the other studies. But again, it's an outdoor study. There are other variables that are affecting. But yeah, but in the end, we found an increase of 32% more trichome density, but this was not correlated to cannabinoid content. So again, this is one study. There are few, but we need more studies to uncover the effects of methyl jasmonate on cannabis because it makes sense. Again, you artificially create that signal to the plant, and in theory, when the plant is under stress or when the plant feels that it's under attack, it would promote the production of trichomes or the formation of trichomes. So, but you need more study because then it can be cultivar related, other outdoor variables, and even concentration rate, application rate. Of course, concentration and application rate, the application method as well, like how you spreading that methyl jasmonate throughout the whole plant. What is the best timing for during the flowering cycle to apply methyl jasmonate? So there are so many other variables
Mhm.
that I'm always hesitant to say, oh, no, methyl jasmonate is not increasing cannabinoid, or methyl jasmonate is just increasing trichome density. So
Yeah.
again, as
Yeah.
we talk, we need to be careful with make some strong statements.
Yeah, yeah. Well, it's all, I mean, it's just a really cool piece of research. And if I'm hearing you correctly, what you're saying is the trichomes are a plant defense mechanism. And if the plant senses some sort of stress, say it's a pest or something like that, methyl jasmonate can be the mechanism through which it receives this signal and then produces a lot more trichomes as part of the defenses. And this is why you'll see that density go up so significantly. And the idea with what the study you were doing was we're just going to take this and we're going to apply it directly without having created the actual stress that would normally produce this compound. And then, as expected, the trichome density goes up pretty significantly as if the plant was under attack, under some sort of stress, but doesn't really increase the cannabinoid percentage within the specific context of the parameters of how it was used, like dose, timing, cultivar, all of that. So I think that's going to be a little counterintuitive to a lot of people who are just going to think intuitively that if your trichome density goes up, that the cannabinoid percentage will go up along with it. So why is it that those things are not associated linearly like we might intuitively expect?
Good question. And the thing is, when we start releasing these studies, and even before releasing this study, publishing these studies, I was talking with colleagues that are working with cannabis and trichome density, and many of them, they're like, yeah, it's happening. It's not really a direct correlation. So we are struggling to see that directly correlation. And then, as you know, a lot of people don't like it. And again, if it was not my lab conducting this the study, I was like, yeah, I would be skeptical. And it was like, it makes total sense. More trichomes, more cannabinoids. And then when I started receiving a lot of backlash about, oh, you don't know what you're doing, this doesn't make any sense. But again, I have other colleagues, other scientists backing up that statement or that confirmation that is not really that directly correlation. So then we decide to go deep on it and then start exploring like the reasons why trichome density alone is not the best correlation with cannabinoid content. And then we publish this other paper. It's a review where we are exploring the flaw, not the flaws, but the things that could be improved in trichome density counting. And then we found that when you're counting trichomes, you're basically just counting numbers. And I'm talking about glandular trichomes. Like we are not even counting the non-glandular trichomes because they do not have any cannabinoid in it. So when you're only counting the glandular trichomes, you're counting numbers. For example, if you're counting 10 very big trichomes and then 20 very small trichomes, they might be the same. But when you say 20 trichomes, you expect to have more cannabinoid, but you're not accounting to the trichome head size. How big is a trichome head?
So if I'm understanding, you're saying like there's a given mass of cannabinoids, and that mass in a certain area could be divided over 100 trichomes, granular trichomes, or 10.
Yes.
And it's the same mass either way. It's just like, do you have way fewer, like less density of much larger trichomes? Yes. Okay. Yeah. Yes. So that makes perfect sense.
But this is one point. Another point that must be considered is when you're counting trichomes, you're not counting the concentration of cannabinoid inside the trichome head. So one very concentrated trichomes can be equal to 5 less concentrated trichome head. So
you're saying that like within a given mass, say there's like just for example, like a milligram of trichome heads, the percentage of that milligram that's actual cannabinoids can vary pretty widely. There could be other components that aren't cannabinoids that could make up that mass.
Yes. And then as you're not considering that concentration, and then you're trying to correlate the number with just numbers, then when you get the HPLC analysis, it's like, no, that's not there. Another thing that should be considered is the part of the plant that you're counting the trichomes. Mm-hmm. So we use the brackets because they're very consistent. So the bracket is a small part, is a special leaf of the protecting the ovary, and it's not very big, and though it's very consistent. So it's an easy and a very straightforward method. Like when you're just select, okay, we are doing trichome density in the bract. But the thing is, you could be using other parts of the plant, the sugar leaf, or even if you're using the bract of the top part of the plant, or if you use the bract of the lower part of the
so there
the
could
plant.
be differential effects potentially between different locations on the plant. When you're doing your HPLC, you're getting a sort of global average of all of the material that goes in there, but the distribution between the different parts of the plant could be pretty significantly varied.
Yeah, or even if then let's say you're just comparing exactly the same part. Like usually when you run cannabinoid analysis, we use the main cola, the top part, because it's the max or the highest yield you can get. And usually in most of the cases, like even if you're just using, like let's say you're taking 12 inches of the main cola, and in that way 12 inches you take a subsample, but to run to count trichomes, and then you take another subsample for the cannabinoid analysis. But one is in the top 3 inches and the other sample is in the bottom 10 inches. Even in that same main cola, the cannabinoid levels can vary. So, and then imagine what could be if you're just using the top flower and the bottom flower. So the cannabinoid is also can vary. So again, that's why we start seeing that it's not that direct correlation. We can see a trend. Sometimes we can see a trend, but I guess it's to consider trichome density as a only parameter to correlate to cannabinoid analysis, to cannabinoid concentration, is not the best parameter.
Yeah. It's
So
not necessarily a very consistent predictor because of the ways that these other things that mathematically will influence the end result can influence it.
yeah, and I know the legacy they like, and it makes sense. Again, things might change, but or because maybe we can aprimorate or improve the way we're counting the trichomes, taking into account other important factors, and then the correlation can increase. So that's why we still collecting trichome density because it's an important parameter, but we use that more as a aesthetics parameter, because
Right.
these you cannot negate. Higher trichome density is correlated to frostier bud.
Right. And so like in the sales environment, still a lot of
It's very
people
important.
really care about the bag appeal, right? They want it
Yes.
to look like it's just caked with trichomes. So maybe that's still a pretty big win when it comes down
Yes.
to quality metrics, because the aesthetics, you get like, you have the same total cannabinoids distributed over more trichomes, but it changes the look in a way that improves the value, even though the potency measurement's the same.
Yes. So I think it could be more quality parameter than a potency parameter, which is still important. And obviously, when you're writing a scientific paper, you cannot write this treatment is faster than this other treatment, but this treatment has a higher trichome density, is more scientifically appropriate.
Yeah. And I think if I remember correctly, in that study, you guys used a fairly aggressive dosing range when applying the methyl jasmonate relative to some of the other research. Am I remembering that correctly, or?
There's one other methyl jasmonate study test, and then they actually saw an increase in cannabinoid. It was funny because they did not see increase in trichome density, but more cannabinoid. Yes, and we found more trichome density and no effects in cannabinoid. So again, this is this is science. So
I love it. I love it.
yeah.
Yeah, it's just like we're gonna run these experiments. They're gonna have a little bit different parameters. We're gonna get different results, and then over time we reconcile it by figuring out what's responsible for those differing results.
Yeah, and again, but this is science. This is what's happening for corn and soybeans and wheat. So we have a lot of studies that all these universities and scientists are conducting for years, and then some are following a similar trend, some are following a different trend. So then you have all a very like a variety of results. But for cannabis, we don't have much yet. We are just creating the baseline right now to support the coming studies.
Yeah, absolutely. So switching gears a little bit, you had a really interesting finding from one of your papers that looked at mother plants, and you had two, I think, cherry wine clones from different sources that responded in opposite directions to identical treatments. What does that mean? How do you interpret that? Like, why is it possible that that could happen with the same cultivar?
Yeah, it's just to, I think we use like 1 micromole for methyl jasmonating. They studied that.
That's right. Yeah.
And then we saw from others study. But yeah, talking about the genetic, I guess the genetic stability for cannabis is something that it's improving. When you talk about seeds and then you compare the technology for that we have for, like, for example, for like corn hybrids. You see, I'm here in Illinois. You go outdoor, you go outside. Now they're planting, or they'll be planting soon. You see that like they're all using seeds, and then you see the laser level. All the plants are identical. So for cannabis, I know the technology is improving. So, but we still rely on clones. So we have our mother plants. Most of the commercial facilities, they're taking care. They have their propagation room. And then basically the propagation is based on clones. The seeds for propagation, I don't know. I mean, what do you think? Do you think in 10 years we will have just seed propagation? We will not be using clones? What do you think? What is your opinion about seed and clones for the future?
Yeah, I mean it's hard to say because I actually thought that we would have been using more seeds by now
Hmm.
anyway. I know there's a lot of research and a few people that I know personally who are working on creating seeds that were genetically identical in the way that clones were so that you could remove a lot of the variability in cultivation. And I haven't checked in on that to see how it's been going in a little while,
Hmm.
but there's certainly some major advantages to using seeds. Although at this point, either because the technology hasn't got there for producing consistent enough ones for like CEA or indoor cultivation, or just because the practices are sometimes sticky and change slowly.
Hmm.
Right now, it does seem like everybody is mostly for the indoor side, especially using almost exclusively clones.
Yeah, and it's working, right? It's not that, oh, this is actually, we are doing it, but it's not really good. It works. But yeah, I get that's another controversial conversation. Is like when seed will be dominating the market, when clones will be the method or the approach for propagating cannabis. So I guess it was progress in your lab because in the very beginning, when in 2021, when I was establishing our lab, we did not have like a mother plant room. We were not just establishing the lab. So basically, what we were doing was just buying clones or rooted cuttings or just cuttings, and and then propagating and using for research. And then we start realizing that, okay, then that's the progression, that's the step. Realize that the clones are not very stable. We already knew that we could not use seeds because it would be a scientifically mess, because there's not genetic stability in seeds. Back in 2021, it's still now still it's hard to ensure genetic stability in seeds. So we decided to use clones, but even the clones, they're not really very stable. So we start seeing some too much variability to the clones. And then, especially from one year to another, why? How
Mhm.
come in 1 year this high CBD clone is producing 8% CBD, and next year it's producing 17 or no or 16? So is it the same genetic? So okay, then we realize that, oh no, buying clones from commercial facilities, I'm not talking about for commercial purpose, but for scientific purpose, it was not convenient because the genetic variability. So then we decided to start maintaining our mother plants. So now we have our mother plants room, we have a propagation room, and we maintain mother plants, let's say 5 cultivars all the time, 24/7, all year long. So it doesn't matter if it's Christmas or summer, we always have a students maintaining the mother plants. So
Mhm.
then, okay, then we move from buying clones to maintain our mother plants.
Yeah.
Then we start using more than one mother plant sisters for the same study. So we thought that, okay, both plants are sisters, they are basically identical. So if we use clones from two mother plants, these will not create any genetic effect. These would not actually be a factor. But then
Wait,
we
just to
found,
be clear, were those two mothers, were they created from like the same original mother?
yes,
They were.
basically
Okay. So,
sisters
okay, cool. So yeah, so they are genetically identical, at least in the genome itself.
in theory.
Hmm.
Yes. So, and then that's our assumption would be, okay, mother plants would not be a factor. So we are not really considering that a factor when we start running the analysis for that study. But then when it's okay, wait a minute, let's separate that. And then it was interesting because a lot of our treatments came back not statistically significant, but the most significant effect was the two mother plants. And we were like what is going on? Like, how we going to justify that these mother plants are not really identical, or not as identical as we thought? So, and then we started exploring, and then we found that even the light intensity, if one mother plant is in one corner of the mother plant room or the chamber or the tent, or if it's closer to the humidifier, or so these can create. And even if you're maintaining our mother plants pretty much. Of course, it's based on cultivar, but the fertilization are the same for each cultivar. Even if the fertilization, the fertigation is not precisely identical, it can also change. So again, we tried to figure out why it was not that identical. And we found that there are some reasons that could be creating this epigenetics effect that is coming from the mother plant, the two different mother plants. So then after we got it, and again, those are the results. We ran the analysis and it came back to the two mother plants. So then after that, then we decided now just one mother plant. We cannot even have that as a factor. It's just all cuttings from one mother plant. So
Yeah, I
but
mean,
that was interesting.
on the commercial side, everybody has the experience of running a room full of plants, and they're all the exact same strain. You make them from your in-house mothers, your team makes them, they're all exactly the same. And there's always a few plants, what we call the hero plants in that room, which if you look at the variability and you plant your yield or something like that on a bell curve, there's always a few that are the extreme outliers in either direction. And some of that can be attributed to like the specifics of like the health of the plant along the way, or theoretically it could be like where it was taken from the mother, how healthy the mother was. But everybody has this experience of looking at a room full of plants, seeing one or two that are clearly the extreme end of the bell curve for production, and just being like, well, if only we could do this for the entire broom, then we'd be absolutely killing it.
So yeah, and then the thing, this is actually very interesting because you call the heroes or the lucky ones, and you see that in commercial facilities. And the thing is, imagine that, okay, in commercial facilities, most of the climate control and the environmental conditions are optimal and the same. So then imagine when in my lab, you're pushing the plants to the limit, like water depth stress, temperature stress, or light intensity stress. So the conditions are not the same. So we're pushing the clones coming from different mother plants to the limit. So then I guess is one more, or even treating with biostimulants or no biostimulants, or even nutrient deficiency or no nutrient deficiency. So then you can see even more these genetic differences, or how the plants are expressing or not, or phenotyping in a different way. So that's why I guess we see even more than in the commercial facility, because in commercial facility everything is standardized. But when you're pushing, then the plants will express the genes in a different way.
Yeah, you're trying to maximize variation for measurability reasons,
Yes.
and we're trying to minimize it when we're trying to do it for production reasons, which makes a lot of sense there. I mean, one of the things that we always do on the commercial side of things, which is when say you have a bunch of plants that are your genetic stock and you have to make your new round of mothers. One of the things that's really important, I think, is to always take the very healthiest clones to use as your new mothers. Never push those into production. You always want to use the absolute healthiest, fastest rooting clones from those mothers to become the new generation of mothers, because almost everybody at some point in time has done the opposite, where they take their best cuttings, use them in production, and then use what's left over a little bit slower for their next round of moms. And if that happens, you kind of get this slow downward slide in productivity over time, where each harvest going forward becomes a little bit less productive than the one before. And there's always, nobody knows exactly what's going on here. We've for years hypothesized, is it epigenetics? Is it some sort of pathogen load that's really hard to measure? There's all these things that are difficult to measure. As a little brief tangent, one of my favorite ways of explaining how powerful epigenetics can be to people is to look up what happens when you take a domesticated pig and then release it into the wild for a while, like a group of domesticated pigs. And in very short order, they start to develop thickened skin, and tusks will develop, and long hair. And these are all environmental reactions. As the environment provides signals to the biology, the way that the genes are being activated or suppressed slowly changes to adapt the animal to that particular environment better. And it can happen like pretty surprisingly quickly. So it's no shock at all that we would see similar things happen in cannabis. And I always wonder, like, what are the epigenetic effects of things like running really elevated CO2 levels generation after generation? That's a situation that at this era of geologic history, a plant's not going to see outdoors, is CO2 levels in the 1200 to 1500 ppm range. And what's going to be happening epigenetically as multiple generations of plants are sort of living in an environment where that's normal, and all of the other potential variations that we have with controlled environment. And as of yet, these are all just things that we get to hypothesize about and don't have much measured data on what's actually happening. But it is really interesting.
And cannabis being the model plant for that, because for some people, not a CO2 enriched production condition or growth is stress. So, and then cannabis is being the model plant for that, because for any other plant that, especially if you're growing outdoors, conventional farming, the 400, 500 ppm, it doesn't mean that it's stress. But for some people, if it's not enriched, if the CO2 is not increased, they see it as a stress environment. So, and again, cannabis is being the model plant for that, like how that environment is triggering the expression of some specific genes.
Yeah. Well, this is a really good opportunity to kind of shift into another part of your research, which was looking at your water stress review. It describes a metabolic switch where restricted water redirects plant energy into cannabinoid production. And there's a striking timing finding in there as well. Late flower stress boosted cannabinoids without hurting the yield, but early flower stress caused really large drops in THC and CBD, if I'm remembering correctly. So kind of walk us through what's happening there.
Yeah, so now we actually have like 3 water deficit stress studies that we are now analyzing and writing the articles. So I can talk about some data that is in my mind, but it's pretty much the combination of all these studies that is kind of behind the scenes.
Yeah, that'd be great.
So yeah, and I guess the first thing that we can start talking about is like the difference of the terminology: water deficit stress and draw stress. So draw stress is pretty much more like a natural condition that is pretty much like a lack of rain. Water deficit stress is usually something more controlled, that is based on irrigation or fertigation or even indoor production or drip irrigation. So that's a difference. I don't like to use much the term draw stress, because draws is lack of rain, not really a trigger or a controlled stimulus. That's another thing that we can talk about: draw stress, water deficit stress, and stimulus. Because some people, especially the crop steering people and then the dry back people, they don't see it as a stress. They see it as a stimulus. So they don't like to classify that as a stress. And I understand, like they're promoting is a controlled stress or is a controlled stimulus purposely, that you're inducing purposely to increase the production of secondary metabolites. So, okay, then with that, we have 3 different terms, but in the paper that we publish, and then the paper that we are going to be publishing, it's basically we are using the term water deficit stress, because it was actually a stress. It was more than just a stimulus. And we did that because when we start running the studies, we want to address water deficit stress. And then we were stressing the plant like 5 days without irrigation, fertigation. And so we studied the frequency. First, we want to test the frequency. How many frequencies of water deficit stress during the flowering cycle would be the best? And then after having that answer, you want to know what the best timing. So for the first one was like, our hypothesis, 1 water deficit stress frequency is stress, extreme stress would be enough. 2 and 3 would be way too much during the flowering cycle. And then based on that, our second study would be 1 period of water deficit stress during different flowering stages: early flowering, middle flower, and late flowering. So then, okay. And then we also did this extreme water deficit stress, because the first time we ran the study, 5 days without fertigation, all the results come back not statistically significant. It was like nothing. And then my student was like, oh, I actually want to publish something. I want to have some statistically significant results. Then based on that, we was like, okay, let's then address the extreme water depth stress. So then we stress the plants for 10 to 12 days, no fertigation. Then we have some confounding, not really confounding, but some maybe controversial results because the water depth stress did not really increase cannabinoid. So that extreme water depth stress, I'm not comparing that with dryback because I don't want people start talking about saying that I'm saying that the stimulus, the dryback stimulus is not efficient. I'm talking about extreme water depth stress, 10 days without water or 12 days without water. So this was not actually increasing cannabinoid. And then we have some answers for that. And that it's basically when you are stressing the plant, water depth stress is also nutrient deficiency because you're not, it's pretty much 12 days or 10 days without applying nutrient. So that's why the next study and then in the control continue the same fertigation. So for our next study, what we want to do, and I think it's going to be a much better comparison, will be you compare the control that will continue receiving irrigation, not fertigation. So during the time that the plants are under stress, not receiving water and fertilizer, the control will receive just water. So then we'll pretty much filter out the nutrient deficiency from the picture. So that's why that's why the reason of perhaps this extreme water depth stress not really increasing cannabinoid concentration could be lack of nutrients as well. So you could have the stimulus, but if you don't have enough nutrients to help the plant to produce the secondary metabolite, then that's detrimental. Again, then that's why the dryback people, which is an approach that I trust, I think it's an efficient approach. They don't see that as detrimental to the plant because again, it's a different method. They're not, then it's another term, the difference between dry back and dry out. So dry out is pretty much what we are doing. It's stress. And dry back is just reducing the water to a certain levels during the growing cycle. So that's why, I guess, to summarize everything that we're working and the ones that we already published, I think there are many things that we have to consider. One is, depending on the study, the water depth stress is also correlated to nutrient deficiency. Another thing is the method of stress, like how extreme is the stress? Are you literally just stopping irrigation and then it's pretty much dry out for several days? Or are you just decreasing the volume or the water levels in the pot or in the rock roll? And also the method, and then the extreme in the method. So, and also the timing when you're doing that. So there are so many other things that we have to consider, but I still believe that the water depth stimulus or stress can be an efficient signal to trigger higher production of cannabinoid.
Yeah. Yeah. I mean, what you're talking about here just gets at one of the things that's so tricky about this and that I'm explaining all the time, which is that it's really hard to isolate variables.
Yes.
When you run experiments like this, you're trying to see what the effects are of reducing moisture. And of course, nutrients get reduced proportionally at the same time. So you're measuring the combined effect of two different things and then how that interacts with everything else. So yeah, it's really tricky. I think the upcoming research is going to be cool where you have controls to sort of separate out the causation there a little bit and see what is causing what, because that's really interesting. Another one of the things that I see as a problem all the time, and I'm constantly having to do education and disclaimers around, is the way people use words to describe different practices and concepts. And it can be really tricky if one person's talking about a dryback and they're looking at like the difference between their peak water content and their lowest water content. And another person's looking at like their daytime average water content and their lowest content. And it's really important to make sure that everybody's using the same terms or ones that reflect each other. Otherwise, you can take the practices from what one person says and apply them and get totally different results because you were just talking about different things without knowing it. And I see it happen just constantly in cannabis, where people just get mixed up on what exactly they're talking about. I really like the idea too of, and it seems like a lot of your research sort of reflects this, but this idea of thinking about plant inputs and signals and stress and how it changes resource allocation within the plant. At any given point in time, a plant has, just like a human does, you have a zero-sum pool of resources which can be allocated to different types of metabolic processes or storing assimilates or whatever it may be. And I think what we're doing a lot of the time when we're manipulating things in the pursuit of crop steering or something like that is changing resource allocation or attempting to in a more predictable manner than we used to.
Yeah, and again, and that's why that's the beauty of cannabis cultivation, because it's like, let's just consider indoor production, indoor grow. If you do growing, let's say tomatoes, what you care is about biomass and fresh fruit. So you just want optimal levels or optimal growing conditions, because you don't need to trigger any secondary metabolite. You could, because you could have more flavonoid in your tomato, but the consumer is not, there's no label showing the amount of flavonoid. So they just care about the optimal levels. But the beauty, that's why cannabis production is trickier, because it's a fine line between biomass production, but also triggering the production of secondary metabolite. You don't want just the primary metabolism. You want also to trigger the secondary metabolism. So, and that's the fine line between not losing biomass, but also triggering secondary metabolism.
We do the exact same thing at Front Row with the way the fertilizer is set up, where for much of the cultivation cycle, we want to provide ample amounts of nitrogen so that we can get that primary metabolism and let it run at full capacity, you know, taking advantage of all the products of photosynthesis and all of the light that we've got. But then at the very end of the cycle, we'll purposefully scale back on nitrogen pretty significantly so we can put a constraint on the primary metabolism. And then the plant is able to reallocate some of those resources into the secondary metabolic pathways. And just like with water stress, if you did this at the wrong time or too much, you end up having really big drops in yield. So it's about getting the timing and the degree of that stimulus or stress correct.
No, yeah, and that's why, and also it's also cultivar related too. Some cultivars will respond for a specific trigger during specific timing. Other cultivars will be different. So that's why it's really hard to make any strong statements for cannabis. And as you mentioned, it's hard to isolate some variables, because, for example, talking about the water deficit stress, you could, decrease the water deficit stress, and then the nutrient, you can also isolate the nutrient just applying water for the control. However, the plants that are under stress, they might be more susceptible to pathogens. So how you isolate that? So that's why, that's why science is evolving and more studies are coming up, and then we can start clarifying and also learning how to be more precise in the stimulus that you'll be promoting.
Yeah. I wanted to ask you kind of like a couple of open-ended question here. One of them is just like on the practical application side for commercial producers, what do you think is some of the practices or things that are not being taken advantage of that you know about on the academic side or you've seen through your experiments, but have just not made it into the production side of cannabis yet?
This is a tough question, I guess, because the thing is, the fact that legit science in addressing cannabis, and we have this lack of like 90 years that there's no really legit science addressing cannabis, or even the empirical observation that we have. It was really tough because they have to do this research or just getting experience with this plant in these hidden places and basements. So there's so much to explore. So, and again, as I mentioned, that we are pretty much just really the papers that we are published is pretty much just the baseline for the other studies that are coming up. So sometimes being pioneer in this area is tough, but also it's rewarding, because it definitely can affect my citation numbers. There's not really much cannabis studies. Like if I'm publishing something addressing cotton or soybean, there's much the chances for more citations are much higher. So it's hard to answer because there are so much to explain. But definitely, as we talk about seeds, how we can find out ways to increase stability in seeds, then this is not going to be only science, but also policies and regulations to create a way that seeds can be more stable.
Yeah.
Even like talking about stress, like there's so much like how stress can modulate, or stress or stimulus, how we can create a optimal stimulus for increase cannabinoid production or increase secondary metabolite production. Because we know that, okay, that's fine. The genetics are very important, very important. Like if you're not creating any stimulus, you just grow the plant as normal, decent conditions, growing condition, the plant will produce secondary metabolite. It's naturally because of the genetics. But how you can actually maximize that genetic? How can you push that genetic to the genetic potential? How can you push that cultivar to their genetic potential? So what is the trigger? What is the stimulus? Is it water depth stress or dryback? And again, the studies that are showing that water depth stress is not really increasing secondary metabolite. Again, it's not saying that dryback is not increasing secondary metabolite. So is it temperature set? Is VPD? What is it? Can we strategically manage the VPD to increase secondary metabolite? Temperature, different media. So we are just about to start a study with zeolite and chitosan.
Oh yeah,
chitosan. That's another way to create chitosan. Is another way to create that, promote that natural stress signal, or kind of create that fake stress, like showing the plant that they're under stress, but they're not really under stress. So that's another interesting research avenue. We didn't talk much about biostimulants. Biostimulants are also, I want to continue exploring biostimulants, especially coming from conventional farm. Biostimulants is really tough sell for conventional farmers. It's hard to explain and for them to understand that some biostimulants are actually effective. They this is snake oil, this doesn't work. I don't want to spend a lot of money for this application and it's not getting anything back. But I see, like, again, the past 3 biostimulant studies, biostimulants from different sources, like humic acid, seaweed, Capaphytos avarezi, and silica. We got really good results, like really good results under stress, and also not and with no stress. So there's so many research avenues in cannabis that we need to explore, though.
Yeah. So let's finish out one more question. Like, I guess for right now, what research question in cannabis do you really wish that someone would fund that nobody's looking at yet?
I guess particularly in my lab, as I'm getting a lot of students, I'm finding a lot of students that with this interest in core crop steering. I think we should, in general, crop steering, dryback, water depth stress. I think we should have more funding for crop steering. And then again, crop steering is kind of talking about terminology that we see more in cannabis. So that's one more reason why cannabis is, especially in academia, is so cool and so innovative, because it's bringing cannabis can be a model plant for many different approaches, and crop steering is one of the approach that you don't really see much for other plants. So yeah, I think in general terms, I would say more funding for crop steering, because I find a lot of students that are interested in crop steering, and we need more money to be invested in crop steering.
Yeah.
Awesome. And
this
is general. It's very general.
Yeah.
There's so many ways that you can perform crop steering.
Yeah. And I take the really broad view of crop steering, just being like thinking about it as the strategic application of a bunch of different environmental parameters for the plant in the pursuit of generating a specific response at different times, as like the very broad definition. And in that context, something that I like to say is we're all crop steering, whether we know it or not. You're just either doing it badly or better. So it'd be nice to have some research on how to do it better.
Yeah, like irrigation strategies, crop steering.
Exactly.
fertilization regime is a is crop streaming. When you're setting up your VPD, this is crop stream. Everything is crop stream.
Yep, that's exactly what I tell everyone.